So we start. Let me perhaps switch computers now. I have the first speaker is Michale Fee. His talk is titled Electrical Circuit Models of Brain Neurons and Synapses. And so we wanted to start with a talk, with the caveats that Jim expressed a moment ago, with a talk that gives us a sense of how the brain works. And I challenge Michale to give us an equivalent circuit model of the brain, because we electrical engineers like to think that way. We then can think of how to render that through devices and circuits. So Michale is the Glenn and Phyllis Dorfingler Professor in the Department of Brain and Cognitive Sciences, and he's also investigator in the McGovern Institute for Brain Research. He received his PhD in applied physics from Stanford. Before moving to MIT, he was principal investigator in the Biological Computation Research Department at Bell Labs. His lab studies birdsongs in order to understand how the brain learns and generates complex sequential behaviors. Michale, thank you very much.
Thank you, Jesus. So it's great to have a chance to talk to you today. So Jim and Jesus have asked me to give a little bit of a primer, essentially, on how we think about neurons in the brain, what they do, how they work, and give a little bit of sort of mathematical or engineering description of that mental model. Okay, so all right. So as you know, the way that the sort of physicist view, or the simple view of what neurons do, is that they essentially sum up inputs from synaptic inputs from other neurons. So we basically think of a neuron as having a bunch of synapses from input neurons, and essentially the input current to this neuron would be a sum of contributions from each of those input neurons, given by a weight of that representing that synaptic strength times the firing rate of the input, and then you sum up those contributions from all those different input neurons. You can write that sum as a dot product, so we have a vector of weights and a vector of firing rates, and we can write the input current as W dot U. Then we have to think about how that input current affects the firing rate of the output neuron. So this is in what we refer to as a rate model. And so we have some function, essentially, that transforms that input current into an output firing rate, and that's this function f. So we can have different functions that we use for our neurons. So binary threshold neuron is commonly used to build perceptrons, where there is some pattern of inputs that the neuron can then fire for a particular pattern of inputs and not fire for a different set of input patterns. We can also build feature detectors by having a large number of inputs, a set of weights that define some receptive field, if you will, in that input space. And then if we have a threshold linear FI neuron, then we can essentially measure how much the input overlaps with that receptive field. Now, in real life, these are extremely simplified perspectives on what neurons are. In real life, the brain actually has a remarkable diversity of neurons, not just binary threshold neurons or linear neurons. There's a huge diversity in the genes and ion channels that are expressed in different neurons. This is a Tisney representation, a clustering diagram of different gene expression patterns of neurons in just one little brain area, the median eminence. You get large diversities of ion channels in cortical neurons as well. Now, what are all these things? Most of these things, many of these things, are different ion channels that these neurons express. These are little proteins that sit in the membrane and pass currents. Each of these different ion channels passes a different ion. They have, you can see that there's a great diversity in these ion channels that represent or that reflect different time scales, voltage ranges, different patterns of inactivation. So there's a lot of different types of neurons at the molecular level. Also at the morphological level, you see neurons with vastly different dendritic patterns, Purkinje cells, cortical pyramidal neurons, motor neurons. And these neurons have different morphology and different distributions of ion channels. Those two things interact to produce different patterns of electrophysiological properties. So some neurons, when you inject current, fire tonically, generating action potentials that are regular. Others generate bursts. Others generate bursts followed by strong inhibition and so on. So how do we think about these different types of firing patterns? How do we think about how the property of an ion channel would translate into the physiological properties of neurons? So our kind of engineering perspective on how these different components interact to produce firing patterns comes from the work of Hodgkin Huxley in the late 40s and early 50s. We basically have what's called an equivalent circuit model. This was a framework that they developed by studying the squid giant axon, which is a very large axon that carries an action potential from the brain to muscles that drive an escape reflex. It's a very large axon, about 1 millimeter across, so you can actually put wires inside of the axon, inject currents, measure the change the voltage of the axon and measure the currents that flow through the membrane that produce the action potential that then propagates down that axon to drive the escape. So what did they do? They put these wires inside this axon, hyperpolarized, changed the voltage level, and measured currents to understand how that action potential is generated. So let me flesh out a little bit about how we let me just develop this model a little bit. We can imagine a neuron with a current source that allows us to change the voltage inside the cell, and then we measure measure the voltage response of the neuron to that current injection. Remember, neurons have many different kinds of ion channels that look sort of like resistances. We can model this as a capacitor associated with the membrane. The cell membrane, and resistances that model the currents flowing through the ion channels. You can model this electrically using currents through these ion channels represented by this I sub L, a capacitive current. Use Kirchhoff's current law to show the sum of the currents into that node has to equal 0. You can write down simple equations like this that relate capacitive current, ionic current, and injected current, and write down simple differential equations that describe the response of that neuron to, let's say, a current step. So the neuron relaxes, voltage relaxes exponentially to some new equilibrium current. Neurons also have batteries. So neurons have large ion concentration gradients. For example, there's a lot of excess potassium concentration inside the cell. Those ions diffuse out through those ion channels. That produces the concentration gradient produces a flux of ions due to diffusion. These pores are ion selective. So, for example, potassium channels are permeable only to potassium ions. Potassium ions diffuse out. That produces a lower voltage inside, and that voltage gradient is associated with an electric field that produces a drift in the opposite direction. You can find an equilibrium voltage of the cell by summing the drift and diffusion voltage, setting it to 0, and solving for the voltage difference that results in 0 net current flow. And so what you find is that concentration gradients together with ion-specific permeabilities result in a stable voltage offset called the Nernst potential. So you put that together with your in your equivalent circuit. What you find is that voltage, that Nernst potential, produces an offset in the IV curve, which you can model as a battery. So neurons have batteries. You can represent different ion channels with different batteries. So there's a potassium conductance represented by this resistor and battery. There's a sodium conductance. And all of those different ion channels work together to control the membrane potential of the neuron. you can have these ion channels have a lot of different interesting properties. For example, they have voltage dependence, so many ion channels have a charged residue inside of the protein that acts as a voltage sensor that produces a conformational deformation of the protein that controls this ion selective pore. So those conductances are really variable conductances. Those are the knobs that you essentially that the neuron controls to change the potential of the cell. And so, for example, you can generate an action potential by turning on the sodium conductances, which then connect the inside of the cell to that battery and raise the voltage. Then you turn on a potassium conductance, turn off the sodium conductance, and it drives the voltage of the cell to the equilibrium potential of the potassium conductance. Okay, so we have batteries and knobs that allow us to control the voltage inside the cell. Those conductances have interesting temporal dynamics. When you change the potential inside of the cell, those conductances turn on with some time course. Some conductances turn on very slowly, others turn on quickly. Some conductances inactivate once they've turned on, so potassium turns on and stays on. Sodium turns on and inactivates because there's a charged residue on the intracellular side of the protein. It's like a little piece of, a little floppy piece of residue that's charged. When the channel opens, that charged residue kind of gets sucked in and plugs up the pore. Okay, so there's all kinds of interesting biophysics that goes on with these ion channels. And all of those different ion channels have different voltages at which they activate. So this shows the equilibrium open probability of those ion channels as a function of membrane potential. And they turn on sigmoidally. Different ion channels have different voltages at which they turn on, different voltages at which they inactivate, and different time constants that are also a function of voltage. Okay, so each one of these different proteins has a different intracellular machinery that gives it special properties, that different neurons have different complements of these types of ion channels that give unique properties to each one of those neurons. Okay, so that's just what's going on at the soma. There's a lot more to neurons. Neurons have thousands of inputs on these structures called dendrites. So there's the soma right there. That's where all of that sodium potential, the action potential, is generated by those voltage-dependent sodium and potassium conductances. That neuron makes a decision to fire based on inputs into this elaborate structure here called a dendrite. That action potential would then propagate down and contact other neurons and release neurotransmitter on other neurons in a mechanism that I'll describe in a moment. Synapses are very small, so a neuron like this might have a dendritic structure that's several hundred microns long. This is a cerebellar Purkinje neuron. Neurons in the human cortex can be several millimeters long in their dendritic extent. And just to mention, axons can be quite long. The axon of a neuron in your spinal cord that innervates muscles in your feet can have an axon that's a meter long. So neurons get lots of synaptic input. The contact areas are small, about a half micron. Packing density is very high. So in a cubic millimeter of cortex, for example, there are about 10 to the 9 synapses. That means there's a synapse on a 1 micron lattice. Okay. In that 1 millimeter of cortical tissue, there are several kilometers of axon and about a half a kilometer of dendrite. Okay. Cell receives typically 1,000 to 10,000 synapses. Purkinje cells can receive close to 100,000 synapses, and an individual neuron has 4 millimeters of dendrite. Okay. So that's just some numbers on synaptic connectivity. Let me just give you a brief overview of how synapses work. So this is the basic structure of a synapse. There's a presynaptic axon. The action potential will propagate down here. There are synaptic vesicles that contain neurotransmitter that are released onto a postsynaptic terminal. On that dendrite of the postsynaptic neuron, there's quite often a little protrusion called a spine that has a lot of specialized machinery for responding to that synaptic neurotransmitter that's released onto the postsynaptic neuron. So again, this thing is about the wavelength of green light. Okay, so it's tiny. Okay, so here's how it works. An action potential propagates down. That's a voltage pulse that then activates voltage-dependent calcium channels in the presynaptic terminal. Calcium flows in, binds to proteins that are specialized at the membrane here that cause those synaptic vesicles to fuse with the membrane of the presynaptic terminal, releasing neurotransmitter into the synaptic cleft, which is only a few tens of nanometers across, so the diffusion occurs very quickly, binds to neurotransmitter receptors on the postsynaptic membrane. Those neurotransmitter receptors then become conductive and allow the conductance goes up, allowing ions to flow into the postsynaptic neuron. That depolarizes the synapse here. Current flows into the dendrite, and that raises the membrane potential. And then you get a regenerative wave of voltage that then propagates down. Sorry, this is in the dendrite, so you have multiple of these synaptic inputs that in some way add up to a change in the voltage. Once that gets down to the soma, then it can activate that action potential that we described before. Okay, so how do we think about what's the equivalent circuit of a synapse? So that looks like this. Again, that neurotransmitter is binding to ion channels on the membrane of the postsynaptic neuron. It opens up conductance, so just like the sodium channel had a voltage dependence that caused it to open up, neurotransmitter receptors of this sort bind neurotransmitter, and that turns on this conductance. Neurotransmitters, receptors are also ion permeable and often have specific ions that they are permeable to, so they also have this little battery. So when the neurotransmitter binds, it essentially reduces this resistance or increases the conductance and connects the inside of the cell to that battery. Okay, so we can now write down the synaptic current as a conductance times the membrane potential minus that synaptic reversal potential. Okay? All right, so there are many different kinds of synapses. Some synapses, for example, a very common synapse in the brain is what's referred to as a glutamatergic synapse, where glutamate binds to this receptor. Glutamatergic synapses have a reversal potential close to 0, while the cell is normally hyperpolarized around -80 or so. So when that neurotransmitter binds, it causes the voltage of the cell to go up, and that acts like an excitatory input to the neuron. On the other hand, there are other inhibitory synapses that are chloride channels. So when GABA binds to a GABAergic GABA receptor, it opens up a chloride channel, and chloride ions flow into the cell, and that lowers the potential of the cell and acts as an inhibitory input because it moves the voltage of the cell away from the voltages at which it's likely to spike. Okay, so there's a huge diversity of neurotransmitter receptors that have different ion permeabilities, producing excitatory and inhibitory synapses. Some synapses have a voltage dependence, so they'll only open if the cell is already a bit depolarized, and they'll stay closed if the cell is at a very negative voltage. Okay, so there are complex coincidence detection capabilities in synapses like this. And they also have different time constants. Some synapses produce a current that turns on very quickly, and others take 100 milliseconds or more to open up. Now, when you think about synaptic inputs, you have to think about the fact that all these synapses aren't just landing on the soma, they land onto this dendrite. And so we often think about cells as having multiple compartments. So we often think of a somatic compartment where this action potential is generated, a dendritic compartment where the synaptic input comes in. We can model those different compartments as little RC circuits connected to each other by resistors through the dendrite, and then synaptic inputs land onto specific compartments. So just as a little piece of circuitry would have an input that lands on like an IC would have an input that lands on a particular pin, inputs to neurons are not all the same. These inputs, these synapses do different things depending on where they land on these different compartments of a neuron. One interesting aspect of this kind of compartmental design like this is that synaptic inputs onto a compartment like this don't add linearly. The effect, the voltage change in the postsynaptic neuron doesn't just keep going up as you give more inputs. In fact, what happens is that as the voltage, as one input drives the voltage of the cell toward this reversal potential, additional inputs increase the conductance, but that doesn't change the voltage because the cell is already at the voltage of the battery. So adding more conductance doesn't change anything. So you can get synaptic saturation, and it's been shown that computationally that produces an OR operation. And there are examples where that OR operation is very useful computationally in the brain. For example, it's been shown in the auditory system that that's a mechanism for noise reduction of auditory inputs into a sound localization circuit. On the other hand, you have synaptic saturation like this, but on the other hand, dendrites often have active conductances. So some dendrites, when you put voltage into them, they don't respond much, and then you put more conductance in, and as a function of conductance, you'll see that suddenly the response in that dendrite will jump up as a result of these regenerative conductances in the dendrite. So these would be produced by voltage-dependent calcium channels, for example. And you can see that if you add multiple inputs to a single branch of a dendrite, you get this nonlinear effect, but if you add the same total input to two different branches, you get a linear summation of those inputs. So where inputs land on neurons actually matters. Now that's actually very important because there is a lot of specificity in the way neurons connect to each other in the brain. So this shows an example of different patterns of connectivity between specific populations of interneurons in cortex, for example, onto pyramidal neurons, which are the principal excitatory neurons in cortex. So you have chandelier cells that connect very selectively to the part of the soma that generates an action potential. And so these interneurons are actually able to veto the output of a neuron. Even though the neuron has made a decision to spike, these inhibitory neurons can just shut that output down. Basket cells, other types of inhibitory neurons project selectively. So here's basket cell projecting to the soma. Other inhibitory neurons suppress other specific compartments of the neuron and implement functions that we don't really understand yet what they're doing. I just want to highlight this idea that inputs into different parts of neurons do different things. This is interesting work from the crayfish by Vu and Krasny showing that inhibitory inputs to the soma actually prevent these neurons from spiking. You can see basically that as you increase the inhibition to the soma, it suppresses the response of the neuron in a way that no matter how much excitatory input you put in, it can't overcome. So this is another example of this vetoing ability, whereas inhibitory inputs into the dendrite, where all these other excitatory inputs are coming, simply shifts the threshold of the neuron. Okay, so you can have really interesting sort of modulatory changes in the excitability of the neuron, or this kind of veto power. Okay, so all right, so I'm going to wrap it up there. But I want to leave you with the thought that neurons are not just points that either spike or not. They don't just sum up their inputs with a dot product. They have a lot of complex and beautiful machinery in the form of these different kinds of ion channels that have subtly different properties that the neurons use in conjunction with their different morphologies to produce complex dynamics in the firing patterns of these neurons that presumably interact with temporal structure in the input to control what the computation is that's being done. Okay, I'll stop there and take any questions.
Yes.
So one of the challenges is that everybody believes that, okay, we got this low power brain, and our current CMOS compared to that seems to be much more power hungry. So, I mean, I know you say here nobody knows how it works, but give any insight into how complexity leads to low power?
Well, so the way that I think about this is along the following lines. So I think that Carver Mead and others in his wake, like Rahul Sarpishkar, have really driven home to me in conversations with them that you really want to do computation on the intrinsic physics of a device. Okay, that's how you save power. You use the intrinsic properties of the sort of smallest elements in order to do computations like this. You use saturation to sublinear compressive nonlinearity to do things. You use expansive nonlinearities in the components to do other things. You use oscillations that are generated by a few molecules of particular types to do something else. So build your computation on using the biophysics of the sort of basic, most fundamental elements in your circuit. I'm not sure if that answers your question.
I'm just trying to figure out why is this low power? I mean, ions, is it something, or ions, lower power, and what's exactly the nature of our ability to do something with lower power?
Yeah. Well, so one of these little devices here is producing an oscillation with a few thousand molecules that are driving current, changing the voltage. That voltage change is feeding back to the conformation of that molecule, changing the conductance, and so you have this. You have this little device with just a few thousand molecules that's generating some interesting dynamics that are useful to the computation. That process is driven by these concentration gradients, and very small numbers of ions moving across the membrane produce large changes in the voltage of these cells because they have a very small capacitance. So you've got small numbers of ions moving in this device, changing the voltage and driving interesting dynamics, and then you have to pump those ions back across the membrane, but it's small numbers of ions that generate these properties. And also, you've got these, each one of those different ion channels has different voltage and voltage dependence and time course, and you could sort of modularly, nature has taken those and kind of modularly plunked those things in different places to produce interesting dynamics. That's the way I visualize it in my mind.
Yeah, question back there.
So, you try to quantify the current. I mean, the voltage is only like 30 millivolts, so that's more voltage. But what about the current? Is that like nanoamps?
Yeah. A nanoamp would be a pretty big current for a neuron. It would make it, the input that would drive a neuron to spike like this would could be 100 picoamps or something.
So there are other projects because of biology, things like Connectome project. They're trying
Yes.
to map the entire being 6 billion of neurons.
Yes.
Interconnection, I'm understanding that. What you're presenting here is dendrites and
Yeah, it's the,
Axons.
I haven't even gotten into the wiring. This is
I was
just sort of the
wondering if it's connectivity of movement of the ions that can interpret it at the higher scale, based on what you're saying. Is there a correlation there between the whole macro and how these ions move at the cellular level, so that we can use that information to understand learning and other features?
yeah.
Has this been figured out yet?
Yeah, I don't know. Maybe I'm not quite understanding the question, but you know, these little elements sit in the context of a much bigger circuit, and there are you know, these sort of the sort of feedback and dynamics also at the circuit level as well that I haven't even touched on yet. So I'm not sure how to answer your question, but these are the parts that you should think of as being the elements of those complex networks in the brain, and they drive and change the properties of those networks in ways that you wouldn't get with just feed forward, you know, a layer of firing rates, dot product layer of firing rates, and so on.
Question over here. Yep.
I'm wondering if you consider such complexity to use the language of differential equation. Will this lead to some differential equation system which is pretty steady, expand along the time scale? And I don't know, it seems that right now the state for these is hard to solve. So my question is, do you think such language is the right direction to trace such neural firing network? And if that it is, how we save power in the future?
Mm-hmm. Yeah, you've raised an interesting question about how you solve this. So nature is kind of put in a nice separation of timescales. So the events that this neuron generates, the action potential here that this neuron generates is very short. It's typically much less, you know, less than a millisecond. That propagates down to other neurons and generates a pulse of input. These other mechanisms of integration in the dendrites and the time constants of the neuron and so on are much slower than that, the time constant of the action potential. So mentally, you know, those of us who study neural circuits kind of separate the slower timescales of these integrative mechanisms and the fast timescales of these communication pulses. So I'm not sure if that was exactly your question, but that's kind of the way we think about it. Other neurons, so for example, in small animals such as C. elegans, these neurons don't even spike. They communicate to each other synaptically through non-spiking mechanisms and get very interesting oscillatory dynamics, and you know, of course they generate a whole brain with only 304 neurons.
Michael, thank you very much. This was a very enlightening talk.
Thank you.
We move on. Michael will be around to